In the last video, we left Cambridge with Charles Darwin was coming back from the Galapagos and was trying to understand how species change over time. And today, more than 100 years later, we are back in Cambridge again, but now with a different group of scientists. These three people, James Watson, Francis Crick, and Rosalind Franklin, are the center of one of the most important scientific stories of the 20th century. I'm talking about the discovery of the structure of the DNA. In this video, I want to use their story as a bridge, first from evolution to DNA, and then from DNA to something that we use in computer science and machine learning, that is genetic algorithms. Okay, so let's start from the two gentlemen on the left. In the early 1950s, James Watson and Francis Crick were working at the Cavendish lab in Cambridge, and they were trying to understand the molecular structure of the DNA. Now, they knew that the DNA had to carry genetic information, and it had to be the thing that stores inheritance from one generation to the next. But nobody knew what it looked like in three dimension. Now, they had this idea that DNA may form a kind of twisted ladder, a sort of double helix, but they needed more than just this sort of intuition. They needed some form of hard evidence. Now, there is also a more informal side of the story, because Watson and Crick often had lunch and discussions at a local pub in Cambridge called the Eagle. And this is the place where Crick later stood up and said that they had discovered the secret of life. Now, many years later, when I was working in Cambridge as a researcher, I used to go to the same pub and even sit in the same area where they were meeting. So for me, this story is not just an abstract story. It's literally the same city, the same streets, the same pub, and so on. Okay, so let's go back now to Rosalind Franklin. She was working at King's College in London, and she was an expert in X-ray crystallography. That is a technique that lets you infer the 3D structure of molecules from diffraction patterns. Now, one of her images, the famous photo 51, showed a very clear X-shaped pattern. Now, back in the day, for people who knew how to read it, that pattern was a very huge clue that DNA formed a sort of helix. Now, there has been a lot of debate about how exactly Watson and Crick saw her data and how much credit she received. But scientifically speaking, what matters for us today is that the work of Franklin, Watson, Crick, and many others together led to the double helix model of DNA that was published in 1953. Okay, but so how does this connect to algorithms and to machine learning? Now, the idea here is that DNA is not just a molecule, is a code. So if we go down here, you can see what I'm talking about. So DNA is a long sequence of 4 bases that are A, T, C, and G. Now, the order of these bases forms a sequence. This is like a long string of these bases that like a sort of alphabet made of 4 letters. Now, section of these sequences represent genes. And genes are grouped into chromosomes. So we can think of a chromosome as a big string of information that we can cut into smaller chunks, and we can call these smaller chunks genes. Now, life is going to use this code in a very clever way. The DNA sequence is going to be copied. Sometimes there are some form of small mistakes, small errors due to mutations, and these changes are going to have an effect on the organism. Now, the environment is the one that is going to test each one of these organisms, and only some of them are going to survive and to reproduce. And this is, of course, what we call natural selection. Now, from Darwin, we had the idea of selection and evolution, and now today from Watson, Crick, and Franklin, and many others, we have a physical mechanism, the DNA, that is a code that can be copied, mutated, recombined, and so on. So keep this picture in mind, a population of organisms, each one with chromosomes that store information and each one tested by the environment. Now we are going now to copy this idea into an algorithm. Before doing that, we have to talk a little bit about the history of genetic algorithms. So most of the things that we have been talking about so far happened in between the 50s and the 70s. In particular, in 1954, Barricelli, that was a Norwegian Italian mathematician, published what is probably one of the first examples of a digital evolutionary simulation. In particular, he used a computer to let simple digital organisms compete, reproduce, and evolve over time. Now, we don't have time to go into details about Barricelli's work, but it is good to know that the seeds of evolutionary algorithms were already there in the 50s. Then in the 60s and 70s, Rochenberg and Schwefel, I hope that I pronounce it correctly, they worked on evolution strategies to solve engineering problems. And this was another step towards what we call genetic algorithms. But it was only in 1975 that John Holland published the book Adaptation in Natural and Artificial Systems, that the term genetic algorithms started to be used. Of course, Holland did not invent every single idea in this field, but by publishing this book, he gave a clear framework, and he contributed to popularize a lot the term genetic algorithms. Okay, now in a previous video we saw this big diagram of this bigger family of evolutionary methods, and we say that genetic algorithms are one branch inside this bigger group, bigger family. Now today we are going to zoom in on that branch and we are going to focus only on genetic algorithms. So let's start by describing both the biology terms and the genetic algorithm terms. We are going to compare them and we are going to see how they change. What's the difference between the two? Okay, in biology we have a population of organisms, right, living beings. While in genetic algorithms we have a population of solutions to a particular problem. Then in biology we have chromosomes that are part of the DNA, as we said before, while in genetic algorithms we have chromosomes that encode solutions to the particular problem that we are trying to solve. In biology we also have genes that are made of the 4 bases, while in genetic algorithms we have genes that are made of bits and numbers or symbols. And this depends on the problem that you're trying to solve. Some cases you may have bits, some cases you may have numbers, some cases you may have symbols. Then in biology we have mutation and recombination, while in genetic algorithms we have mutation, crossover, the different operators at our disposal that allows us to generate new candidate solutions. And then in biology, of course, we have the fundamental mechanism of natural selection. I've been talking about this in the previous video. And in genetic algorithms, instead, we have a fitness function that allows us to estimate a fitness value that we can then use to evaluate the various organisms. In the previous video, we also talked about this basic loop of genetic algorithms, and this, of course, is fundamental for the functioning of the entire method. We start with a population of candidate solutions that typically can be randomly sampled from the solution space. And for each one of them, we evaluate a fitness score that is going to represent the score of that particular solution. So we have a fitness value for each candidate. Then, based on the fitness score, we can select the best individuals, and we can use the best individual to generate a new set of solutions candidates that will represent the new generation. And this can be done by using mutation, recombination, crossover, so on, to generate this new generation of candidates. And then we start from scratch, right? So this is a loop. We're going to repeat this for many generations, and this will give us some improvements, right? So at every generation, we are going to get a new set of candidates that are going to be better. They are going to have a higher fitness. Okay. So let's try to go over a tiny intuitive example of what's happening. So here we have two axes in our Cartesian plane. We have the fitness in this axis, and here we're going to have the solutions. Okay. So now we can draw a fitness function. It could be a complicated function like this one. And we have to assume that we don't have access to this function. We don't have access to this formula, to this equation. We can just estimate this function at single points. So now imagine to have a set of candidate solutions on this axis, on the horizontal axis. And these solutions, of course, they are going to have a specific fitness value. So imagine that, for instance, this particular solution is going to have this particular fitness value. Now, at the beginning, we are going to sample a bunch of solutions. Some of them may be better than others. Some of them may be worse. And this, of course, depends on the way you initialize the population. We'll try to spread the population across the solution space to cover different areas of the fitness function. And then what we can do, we can estimate the fitness value for each one of these solutions. For instance, if we can estimate the fitness value for this one, or this one, and so on. Then we're going to select the best candidates. So in this case, we're going to select probably this one on top, this one, right, and possibly this one. We're going to discard the others. And then based on these three, we're going to generate new candidate solutions that are going to be similar to them, right? So probably they're going to fall here, here, and they're going to fall here, here, and maybe here. Of course, crossover and mutation doesn't guarantee that the solution is going to fall close to the parents. So sometimes the solution can fall far apart. Depends from the type of mutation that was applied. Now, the interesting thing here is that as we go from one generation to the other, we will see an improvement in terms of fitness because we are selecting the best solution, so we are using it to generate new candidates. And therefore, we will see that we will get clusters of solutions. For instance, here, or here, and so on. And then it depends how we are going to set the parameters of selection, reproduction, and so on. And in some cases, you can have solutions that are not going to fall inside these two red circles, but they are going to fall outside of it, depending on how much strict we are with elitism parameter. That is basically the parameter that allows you to select the top solutions and basically change the parameters of these top solutions. Okay, now the important bits that you have to understand here, as I said at the beginning, is that you don't have access to the green function. You don't have access to the fitness function. You can just estimate this function at one single point. And this is fundamental because it means that to work with genetic algorithms, you basically don't need to have access to this underlying function. It means that you don't care if the function is differentiable or not. And this is a big advantage for us because majority of modern methods that we use, like deep neural networks, they need to have a differentiable function. Here, this is not a constraint, so we can use these methods also for non-differentiable functions. And in fact, we don't need to have access to the underlying equation that we want to maximize or minimize. In this case, maximize, of course. Okay. So let's try to close the circle here. We started with Darwin and natural selection. We had populations of organisms that change over time. And then we moved to Watson, Crick, Franklin, the equal pap in Cambridge, and the double helix. And we saw that the DNA gave us a sort of clear idea of a code made of chromosomes, genes, sequences that can be copied and mutated. And then we jumped to the 50s, 60s, and 70s, and we saw that there were many scientists, engineers like Barricelli, Holland. They started to use these ideas to solve computer science problems. Now, in the next video, I'm going to dig a bit deeper, explain a bit better what are genetic algorithms. We will see how we can create a chromosome, how we can encode solutions of a problem inside a chromosome, and we'll see how we can define a population, how we can select the best candidates and generate a new generation of candidates, and so on. So, if you like the video, subscribe, give a bit a thumbs up. Also consider joining our Sapiente community. That is a great community where we can share all these ideas. We can talk a little bit more about genetic algorithms, neural networks, and so on. Thank you for watching, and I'll see you to the next one.